Okay, thank you for the introduction. Good afternoon, everyone. So my name is Amos, and I will be talking about paternal preconception plasticizer, authors sperm, methylome, and embryonic development. So I'm from Environmental Health Sciences Department. I'm a PhD candidate from Paternal Lab. So, um, so our research question was to know if preconception phthalate exposure would alter sperm epigenome and embryo development. And so this was based on the understanding that phthalates are a class of endocrine disruptive chemicals. And a very good example is DHP, which is one of the most widely used phthalate compounds. And also that phthalates are widespread in consumer products and are detected in majority of US population. So our previous study from the lab actually showed that in IVF cohorts actually showed that male urinary phthalate metabolite was associated with altered sperm epigenome as well as poor blastocyst quality. So findings from these two studies actually suggest that sperm epigenome may be inherited to influence early life development. And so to corroborate these two studies, we set up an experiment where mature male mice were exposed to DHP 2.5 milligrams per kilogram per day, as well as to control. And then we targeted this level based on the no observable adverse effect level of DHP, which is 4.8 milligrams per kilogram per day in rats. So the exposure went on for 70 days, which is approximately 2 spermatogenic cycle. It is worthy of note at this point that spermatogenic period has been demonstrated to be sensitive to environmental factors and behaviors such as diet, exercise, and even other toxicants. And so following that, we mated the exposed males to unexposed females, and then we collected sperm for DNA methylation analysis. And this was done using RRBS, which is a reduced representation bisulfite sequencing. And then the pregnant females were followed for 7.5 days until development of blastocyst, and then the embryos were collected. And so at this stage, we ran methylation and also gene expression analysis using the RNA-seq technique. And please note that the embryos, have two different types of tissues. There is embryonic tissue which develops into the fetus, and then there is extra embryonic tissue that develops into the placenta. So from here, we observed interestingly that exposure to this level, which is 2.5 milligrams per kilogram per day of DHP preconception, actually altered sperm epigenome at 410 differentially methylated regions in sperm, which is DMRs. And so this resulted in more than 2,000 DMRs in extra embryo and more than 800 DMRs in embryo. It also resulted in 12 differentially expressed genes from RNA-seq analysis, as well as over 1,200 differentially expressed genes from in embryo RNA-seq analysis. Please also note that the DMRs that we observed in F1 tissues were about 2 to 4 foot higher than what we observed in sperm, which was really interesting. And so the next thing we did was to look at the biological function of the differentially expressed genes, which were the 12 and the over 1,200 that we observed. And so to do this, we set up a design, which is an ontology analysis using a dot plot. And so this is the dot plot. On the dot plot, we would see the size of the dot represents enrichment of our genes, while the column dot represent the p-value. So there are two columns here. There's the embryo and the extra embryo column. And so one interesting thing from this enrichment analysis was that our top enriched terms were actually important in pattern specification process, embryo development, and tissue morphogenesis. And so we conclude, and this study again is one of the, is actually the first study to experimentally show that male preconception phthalate alters sperm epigenome and affects early life methylation and gene expression. So our take-home messages from here is that sperm epigenome can transmit environmental information to the next generation, and also that while the burden of reproductive success falls on women, males have environmental responsibility that starts prior to conception. So I'm going to appreciate our lab members which is, these are lab members, as well as the collaborators, Alexandra Suverov and Jesse Major. Thank you. So I'll take your questions from here.
Wonderful. That was an excellent talk. Floor is open for questions.
Hello, you do have a question. So what steps can we take to eliminate the exposures of phthalates in humans?
Daily exposures in humans?
Yeah, of phthalates.
So if we talk about daily exposures of phthalates in human, we know generally that phthalates are mostly found in a consumer product like plastics, even they're using PVCs and other consumer products. So one major one that comes right to mind is avoiding plastics in microwaves because there are phthalates in them, and then avoiding especially in babies, there are plastic bottles, which some of these plastics are made of phthalates. So we want to reduce that, in babies. But there are other ways that we also have this exposure to phthalates, especially in medical equipment, which that may be difficult to actually to reduce, especially children in NICU, which is neonate intensive care unit. In fact, most of children in NICU have actually been shown to have elevated level of phthalates in their blood. So that may be difficult to achieve, but if only some of these plastics, we could have alternatives to them. Although there are alternatives to DHP, which is DINCH, but that is only has been banned in products like baby products and not in other products. So, but at least the little we can do is just to, as adults, to reduce exposure from food and other sources.
Thank you. You have another question. To what degree would a parent's exposure to environmental impact their offspring's sperm epigenome? In other words, could there be an indirect effect passed down from parents that is not caused by phthalate exposure in the pups?
Okay, so let me take the first answer. The first, you said to what degree can the parent exposure impact offspring development on Earth?
Impact their offspring's sperm epigenome.
So that question, so parent exposure to phthalates, this is actually the first study that actually has done that experimental exposure. But there are other exposures that have been shown to impact the offspring epigenome, like diet has been shown to impact offspring epigenome. Diet, even exercise has been shown to impact sperm epigenome and then been transmitted to offspring. So, but in terms of phthalate exposure, this is the first study, and so to what degree we do not know, but that would be a good research question for future studies. And then there is a second question that was also asked. I can't remember.
In other words, could there be an indirect effect passed down from parents that is not caused by phthalate exposure in the pups?
That is not caused by what? I didn't get that, sorry.
That is not, okay, so could there be an indirect effect passed down from parents that is not caused by phthalate exposure in the pups?
Yes, so there could be like indirect exposure, indirect effect, like behavior, like diet. So we've actually seen a study that showed that like in rats or in rodents, where exposure to signals like chemical cues actually was passed down to the offspring. So we can, from there, we can say that behavior like exposure to not direct exposure, indirect exposure, like like exercise, like diet, can actually affect downstream generation.
I think we have about 40 seconds left, Irene. So maybe
there's a few more questions for Oladele. So why are there only 12 differentially expressed genes while thousands of differentially methylated regions?
So again, those questions, so this is the first study. in this area. So we cannot say exactly why. But of course, there could also be other epigenetic mechanisms like histone modification and RNA-based silencing that we haven't explored. So we cannot say exactly why, but of course, future generation, there is a lot of studies that can stem from here about other epigenetic mechanisms to look at why we are seeing some of these effect that we are seeing in DMRs, in methylation versus gene expression.
Great, thank you.